Hello, my name is Samantha Segura, and this is my YouTube project over the research of pesticide-contaminated pollen contributing to colony collapse disorder in honeybees, Apis mellifera. In the year 2006, the U.S. witnessed a large-scale loss of managed honeybee colonies, Apis mellifera, which continued into the year 2008 and declined shortly after. This occurrence was termed colony collapse disorder. Colony collapse disorder, frequently abbreviated as CCD, is known as a phenomenon when all honeybees disappear from the hive, leaving the queen and brood alone, ultimately leading to hive failure. There are many different factors that are presumed to contribute to CCD, such as pesticide exposure, air pollution, viruses, parasites, heavy metals, and more. However, in this presentation, we will be focusing mainly on pesticide exposure. So the main question is, how does pesticide-contaminated pollen contribute to colony collapse disorder of the honeybee Apis mellifera? Well, due to high-quality seed technology, there has been an increase in pesticide development, which leads to greater risk of nectar and pollen contamination. Pesticide contamination has been assumed the most common reason for colony collapse disorder in honeybees. Neonicotinoids is an insecticide known for its toxic effects amongst pollinators, primarily bees. There are certain factors that influence the level of pesticide residue in pollen, and this includes crop-related parameters, discrepancies in pesticide application, such as timing and dose rate, physiochemical properties of the active ingredient, and the environmental conditions. This negatively impacts the honeybees as they only have about half as many detoxifying enzymes compared to insects that are pesticide resistant. This increases their sensitivity to pesticide exposure, further weakening their ability to fight off bacterial and viral infections. Pesticide exposure can lead to delayed development, impairment of the immune system, and a shorter lifespan. Foraging bees that come into contact with contaminated pollen may transfer these impurities to the hive, which could end up infecting the entire colony. There's been a lot of research and studies that have looked into pesticide exposure and honeybee health, relating them to colony collapse disorder. In this study in particular, pesticide residue was collected from pollen, wax, and nectar. Of 161 pesticides, 124 residues appeared in pollen. The top chart illustrates the different types of residues found in pollen, highlighting the quantity and frequency levels of each. Insecticides contaminated pollen the most frequently. Thiamethoxam, a type of insecticide, poses the greatest risk to honeybees when ingesting contaminated pollen. The bottom table lists out the pesticide residues that were commonly found within 1 gram of contaminated pollen. Only 5 compounds show a high risk in pollen residues, which has to be above 0.5%, and this can seriously affect honeybee health. The 5 compounds include thiamethoxam, phosmet, chloropyrifos, imidacloprid, and clothianidin. And I apologize if I butchered any of these names. The table measures the probability and time to reach the lethal dose of honeybees in regards to contact exposure of 1 gram of contaminated pollen. There is sufficient risk to nurse bees and larvae of pollen residues that contain all 5 of these compounds. Of all honeybees in the hives, the main ones responsible for pollen consumption are larvae and nurse bees. The following table highlights the difference in life longevity after contaminated pollen has been ingested. The 3 circled have a shorter lifespan than the majority of the other worker bees. The average residues found in the collected pollen samples approaches an oral lethal dose within the lifespan of the nurse bees and larvae, affecting colony growth long term. The following chart demonstrates a study done on Apis mellifera regarding specific insecticide residues levels found within plant tissue, bee pollen, and more. The median thiamethoxam residue levels in pollen collected from foraging bees was similar to the quantity found in plant tissue. This can be concerning due to the fact that plant tissue can be very absorbent, and pesticide exposure doesn't necessarily mean that the pesticides have to be applied directly onto the crop. Any crop is capable of taking up these chemicals through the soil, through runoff water, and such. So the fact that the pollen is nearly at the same level as the plant tissue is cause for alarm. Neonicotinoids cause toxic effects towards bees and can cause impairment of honeybee homing behavior, foraging behavior, or reducing colony growth and queen production, thus leaving possibility for CCD to occur. Lab research and studies have demonstrated that small amounts of neonicotinoids impact honeybee health, and this includes by disorientation, reduced communication, impaired learning and memory, reduced longevity, and disruption of honeybee brood cycles. These negative impacts have the ability to impair their olfactory learning and memory due to the abnormality in biogenic amines mediated neuronal signaling. Therefore, foragers can struggle to return to their hive. Another possible cause for CCD. The figure highlights studies done in the US concerning colony collapse disorder of Apis mellifera. The events circled all have two things in common: pesticide residue contamination and honeybee colony collapse. I included this image to help support the claim of how pesticide residues can affect honeybees to the point of CCD. The image shows how different types of pesticide residues are absorbed by the honeybee, and then a response is given. The response in this scenario would be the colony collapse disorder. In conclusion, pesticide residue found in pollen does not solely lead to CCD in Apis mellifera. However, is still considered to be one of the major contributors according to several studies and research. Pesticides have long been harming our pollinators, primarily honeybees. Large-scale loss of honeybee colonies will negatively impact the agricultural industry and cost the industry billions of dollars. This would also disrupt our food chain, as we wouldn't have the bees to be able to pollinate our crops. Certain levels of neonicotinoids can lead to these detrimental health effects. Neonicotinoid chemicals are the most frequently detected on pollen. A few ways to help keep honeybees healthy is applying pesticides in the evening when foragers are less likely to be out. Using pesticides appropriately according to the label, as there is a lot of misuse of pesticides, especially with homeowners. Or switching to a less toxic or rapidly degradable pesticide could also be a beneficial alternative. Urge the US EPA to take action against regulating pesticide usage with respect to pollinators. This would also help primarily the honeybees by protecting them. Here are all my references, and thank you for sticking with me through my presentation. Hope you enjoyed.